something wrong with my computer. It takes some time to set it up. Okay, so in this team channel, I have one link to a paper. That paper, we will visit that paper and today to download the data from that real paper. So if you have the channel in there, lecture 3, take a look of that paper published in Nature Communications. While we are waiting for this computer to settle down. So by reading that paper, I want you to find the geodatabase accession number and see that paper provides multiple numbers. What you want to do is to identify the real data they generated by the author of the paper themselves, rather than they cite other data and then to compare with. So that's what I want you to do right now, to visit this link in lecture 3. So this is a study to compare cell population collected from the diseased tissue compared to the healthy tissue. So this is the paper I'm talking about. It's a single cell data analysis. Peroxis resolution in the single cell level, and to compare the lesional tissue with adjacent healthy tissue. So while we are waiting for my computer to set up, you spend some time to read through this paper very quickly and to find the data set that we can download and play with. So we are going to use this data set to see the basic QC we can conducted on this real data set and some principle behind each step. So in the same time, I'm upgrade the toolbox installation. There are a couple of bug fixing in the last week. So if you want to have a latest version, you just run this. From your history of your comment, you should be able to find this line of code. As long as you are in the correct folder, you should have update this folder by itself after you unzip this. Okay, so I'm done from my side. So now I'm going to use a small example to illustrate the basic data structure we are going to put in the toolbox for the analysis downstream. In the middle of this process, we will revisit the distance calculation, the vector manipulation, etc., etc. We discussed in the lecture on Tuesday. So let's see, for single cell data, remember we need two variables to make a compound variable in order to put in the toolbox. The first variable is big X. This is a matrix contain the single cell data, which is a count for each cell and each gene. Let's see, we have a small X, okay? We are going to play with a small toy. And then another variable is G, right? G is a vector of string, which gave you the name of the genes. In this case, we will have 3 genes. If you double quote, put it in gene 1, gene 2, gene 3. So I will type this in MATLAB. So we have 2 variables to start with. So G and X, I will fill in the numbers here. Okay? So in this small case, we only have 3 genes. So this matrix will be 3 by 4, 4 cells. I will define the name of gene first. Okay, this is very easy. So I have a G, small, and then I'm going to define a vector contains 3 elements. So this will be the gene, let's see, just ABC, okay? B. That's it. I can transpose it if I want to be, now it's horizontal, but if I want to vertical, I can do either to do the transposition. Okay, transposed Okay, this will transpose in the vector of the string, or I can do this vectorization. Okay, you don't have to, because it is a list of gene name, doesn't matter. Now I'm going to define this matrix X. Now I gave you a job is to, if you have imaginary data set, say 3 genes expression in 4 cells, how are you going to put those numbers? You can see I can put 1, 2, 3, 4, 5, 6. It's okay, but then if someone is an expert, they will look at it. This is not realistic. So I want you, from your imagination, let's say, I want to put those numbers. I made it up and gave to an expert in the single cell data analysis. Let them to see, okay, they cannot tell whether this is a real number or a number you made up. So we have some principles to see, okay, so number 1 is this has to be non-negative. So you don't put -1 because there are counts, right? So another rule is each row represents 3 genes. If the randomly select 3 genes, you don't expect 3 genes have the same expression level on average. So maybe first gene expression level on average across cells are high, second lower, third maybe even lower or higher. So you want to make some variation. And also for the columns, you also don't want to be the same different cells. You don't want to have a similar variability. So you want to make something that looks real. So that's your job. And we have some general impression of what this data looks like. You should expect some zeros in there, okay, because sparse matrix. So yeah, just go ahead and we type some data in, okay? So the first gene, first cell, maybe 12, 11, and then maybe 4. So I put the integer. I don't want to put 0.5 because this counts, okay? So that's my first gene. You see the expression level on average, it's maybe 8 or 7. Then my second gene, it's a lowly expressed gene, okay? And my third gene could be higher expression, 8. Suddenly you have a 79, it's a really high number. And then you have 1, 0. Okay. This is not real data, but I follow some kind of rule based on my experience, my understanding of the data itself. It could be a big real data and the corner of this looks like that. All right. So next week we are going to discuss in terms of statistics, how can you simulate a huge set of data and make it look like a realistic data set. So this will involve in understanding the distribution, understanding the process of data generation. Maybe you want to simulate using Poisson distribution, different genes have a different average, and the average across genes follow gamma distribution. So a lot of stuff built in there, but for now we just, okay, from our imagination to create something like this artificially. From this, we can see, okay, gene 1 have a higher expression on average when you cross all the cells. Gene 2 certainly is a very low expression gene, only express 1 and 1 in second and the fourth cell. And the gene 3 looks very special. It has a really high number in second cell, but suddenly becomes low in others. So the variability is higher in the third gene in terms of magnitude difference across cells. So these 3 genes I use as a representative makeup data set for you to and appreciate and somehow get some idea of what it's supposed to be, okay? Now I gave you a job is to calculate the average of each gene's expression. The function is called the min. If you put x in there and put 2, tell you the direction. I want to take average by rows. If you don't put anything, it will calculate by columns. Okay, so this gives you the average expression for the 3 genes. Okay. Now I want to compute an index called the library size. So library size, the definition by definition is how many reads or how many UMI counts in each cell in total. So library size is a term inherited from the bulk RNA-seq data analysis, because when you sequence bulk RNA-seq, you not by the cell. So each column here is a cell, but in bulk RNA-seq is by sample. And people will calculate for sample 1, the first column, second column, sample 2, how many reads they sequenced. So how big is the library they sequenced. So it's called the library size. But we use that term to see, okay, in the cell 1, we sequenced, get reads 20, and this is 5, this is 6. So how can you calculate the library size if I gave you the big X matrix? So you know how to calculate average gene expression, right? You want to calculate the library size, which is a summation of the columns numbers. So this is using the function sum and then 1, okay? 1 tell you I want to do the column-wise summation. If you put a 2, you can still calculate, but it's by rows. Those are the basic function you need to know. So if you want to deal with the data yourself rather than using the Excel spreadsheet, the beauty of this is that no matter how big this X is, same function, you get everything, okay? Even it's millions of rows, millions of columns. Are you okay now? So now I'm going to show you how can we combine this X and G into the variable we commonly use in the toolbox, because I combine them. I don't want to deal with them all time independently. I combine them and make it called S single cell experiment. So it's a name, SCE, and it's a variable name for an object. This object contains 2 variables, most essential 2 variables. To do this is you give the name called SCE, that's the object, then you use capital single cell, then you push tab, you should have this. Then you give X as a variable to construct this object. Okay. So here is I show you how to use the raw data. If you manage it to get X in the MATLAB for some some way you type that in, or you read from my file, or you import. Then you have a gene name. Then you can build this object. So once you have this, you can start to use Toolbox to handle, to manipulate everything's content inside here. Okay, any future annotation, cell properties, or other statistics will be inside this one. Right now, this one only contains two matrices, one matrix and one vector. Any questions? Yeah, okay. So you can take a look of this compound variable by type this, and you can see a lot of fields and other pre-prepared data structure, and also pre-defined the variables. So inside this compound variable, you see the properties. So again, here it comes in the object-oriented programming design. Who have idea what is object-oriented program? You know, okay, object-oriented. Google. So this idea is to encapsulate all the information under the single variable and make this as an object. And then for the object, you have properties, you have actions, you have functions to make your coding easier. So when you have a complex structure and you give this as an object, and then you have properties, you have corresponding functions to deal with, to change the properties of the objects. So those are the ideas. Right now, so we have X here and the G here. This is already defined. So three genes, 3x4 data. And this is embedding and randomly generated. So we don't have real embedding, and the C will be category of the indicate of the cell. So you see this like a 3D, this position of each cell. And this is a category for each cell. We are going to label the cells by different colors according to the membership of a C group later. So there are other properties. So with this, and also this report this as an internal properties, so number of cells, number of genes, all the information from your input. Okay, so from this X and G you gave to the object creation function. Now you can start to use this as CE as input for the toolbox. If you don't have an input, the toolbox will find a source to give you the interface to ask you to read a data file or import. But right now, because you gave the input directly, so let SCE tool to visualize, to show you the results. So we only have 4 cells. So this should be giving you only 4 points. Each point is a cell you defined by yourself. Okay? It's hard to see, and there are 3 genes, 4 cells. Okay, that's how basically how the information has been pushed into this interface. Okay. So I click this button to make this clear, and I can change the. Hope I can change the color. Yeah. 4 cells in the 3D space. If you check the first icon here, you will see the number of genes you just gave yourself. Okay. So this is how the information has been passed on into the interface for you to work on. Now we are going to stop using this for a while, and then we go back to the command line so we can treat each cell as a vector or data point to see how can we apply the knowledge we used in the lecture to calculate the distance between cells, okay, using 3D small example. And after that, we will come back to the interface to play with the real data from download from the paper I just shared with you, okay? So now I just close this. I still have my X defined, also my G, which is the name of genes. Now to be clear, if we consider our data point, our information is column-based, so each column is a vector. So this is a 3D data set because we have 1, 2, 3 genes to define the property or position of each vector or each cell in the 3D space. So I can plot, see my first cell should be in X12, Y0, and Z8. So how can I plot the position of the first cell? We need to know how to extract the first point. So this will be the row, first column. So this is 12. And I need to extract the second position, which is first row, second position. This is my second number. The big X, first row, no, wrong. So I'm talking about the first cell, 3 genes, okay? So that's my coordinates for the first cell. They are X, Y, Z coordinates. So I can do the plot 3 to give a visualization of this. Where is it? So I'm going to use a square, the right O, okay? Okay, this is my first cell. It's in the position 1208 in the 3D space. Then I'm going to use a hold on, so to hold my current figure here. So I can start to plot the second point, the second cell in this space. So the second point, I'm going to use blue point, then, so this will be replaced with the second, still first, second column, second column, second column. So this will put another point. This is my second cell, and that's the position in the 3D defined by the 3 genes. So right circle is my first cell here. My second cell is blue circle over there. Now I can do the third cell, which is, let's put blue square, okay? So you know you can do 3, the third, the position. It's hard to see, but because the range of this data is so huge, the third cell has been pushed back to here. The coordinates is this one, okay? And then for the last cell, we can see, okay, number 4 column, number 4, you can have a red square, okay? Now we can do a so-called box on, grade on, okay, that's it. You have 4 points in this 3D space defined by the expression level of 3 genes, all right? Now to make things easier, let's try to see the first cell is x1, small x, okay? The first cell. My second cell is second, x3, x4, now please calculate the distance between first and the second cell. When I saying the distance, if I don't see anything else, the distance means Euclidean distance by default, okay? So I want you to compute the distance from x1 to x2. If you know how to do this, you can do x1 to x3, x1 to x4. You can use whatever method you have. Just let me know the number once you have it. Have you got the number? What's the d, the distance? What's the number? 71? Not so sure. Let's see whether this is true. So we have a number, the 71 by Cousin, and she found. I don't know if it's true or not, but that's by definition, you have to this minus that to take square, this minus that. So this is the classical way, or is formal way to define the distance between these 2 points, right? You use Pythagoras' theorem to find the difference and the summation of square root. That's the distance. I see whether it's 671 or not. Oh, something wrong. Call function user per mismatch. Plus. Yeah, maybe. 71, yeah. 71, 0.1. Okay? Do you get the same number? You know how to do it? Okay. So x1 is this, x2 is this. Yes, the distance is 71. So the easy way is to do norm x1 minus x2. So this is for vector based. So this should be the d. So this is also the same. So now we are doing this vector operation. So you one vector minus another and take the norm. So you get the same results. Okay. So this is Euclidean distance. That's 4. So now you have the, you know how to compute any pairs of distance between data points. You know how to do 1 to 2. You also should know how to do 1 to 3 or 1 to 4, whatever. So let's do a loop so we can compute all the pairwise distance between each pair of them. So I will do iteration to do 1 versus 2, 1 versus 3, 1 versus 4, and then to do 2, 3, 2, 4, and then to fill up a distance matrix. So I will define a distance matrix as a 4 by 4, right? So I just prepare a distance matrix before I start to fill in the values. Now I'm going to build a loop, see, for I equals to 1 to 4, for j equals 1 to 4, I will do the d I, the j position is norm x I row minus x j row and end. Okay, so this is a way to to compute pairwise distance. So this is not the most efficient way to write the code, but just let you easily to understand that this will be 1 to 4. So when I equals 1, I start to loop inside the j. So I is 1, and I do 1, 2, 3, 4 here. So I start to fill in the fourth row in the D. And where I select the column, so that's different cells from cell 1 against cell 1 itself first. Then the norm should be 0. And then second. So this is a loop. You will very easily get a 4x4 distance matrix that's between each pair of the cell. Any questions for this? Okay, in this distance matrix, the position 1, 2 is the distance between cell 1 to cell 2. That's what we just calculated first pair. And then 2 to 1 is symmetric, so the same. Well, the position, the cell from 1 to 3 is 10.63. You have corresponding index of the coordinates of the numbers inside this matrix tell you this distance between which cell to which cell. Is that clear? Okay. Imagine this is not 4 cells, maybe 1 million cells. You can use the same equation to calculate a distance matrix, pairwise distance matrix for 1 million cells. And from here, from this distance matrix, as I said, we can use multi-dimensional scaling to put the cells in different position to reconstruct a map. This map will keep the same pairwise distance between all the cells. But this map will be very useful to tell us which cells are the neighbors to each other, which cells are the outer layers, which cells should be independent clusters. Okay, so that this matrix is important. As long as we have this, we understand the whole structure of data points as a group of cells, right? Now let's compute the cosine distance between x1 and x2. So again, we have x1, x, so we have x1, x2, so please calculate the cosine distance between these two. Using the definition we we gave in the lecture. So what is cosine distance? What's the formula you can use to calculate this? So whether this is the same as a Euclidean distance, you will have 3 minutes to compute cosine distance between these two data points. And let me know once you have the results. Yeah. Mm-hmm. Invalid use. Oh, because you haven't gave the, you only give the index but not the variable.
Oh.
Yeah.
Oh, thank you.
Not the, yeah. So which variable you want to get this? Yeah. So the definition of the cosine distance, so if it's d, this is 1 minus cosine theta. Theta is between the angle between these two, right? So this is 1 minus dot product x1, x2 dot product divided by x1 norm times x2 norm. So we can express this directly in MATLAB. So this is d again is 1 minus dot x1 x2 divided by squared norm x1 times x2. Okay, I just almost directly copied the definition using the language MATLAB. Then you should get the results. Okay, so this is 0.3359. Yeah, I'm trying to use predefined the function called the p distance, see whether I can get the same result. Yeah, let's search how to calculate. So I try to find the answer which has some function has been already prepared for calculate this. Oh, that's the p distance 2. Okay, so I can use p distance 2 cosine. It requires so the p distance requires 2. I believe the horizontal. So you need to put the correct dimension. So this is using a built-in function, the p distance 2, that's calculate pairwise distance between points among two matrices. This is a built-in function, so we don't have to write in this way, but you should get the same result with your definition-based distance. So the d, the same result. Okay. Let's take 5 minutes break. We come back to start to play with real data. Okay. Okay, so this kind of numeric experiment on the computer, and then to match you learned from the theory, it will be very useful if you have some time to play with all this. And for example, you can demonstrate why this cosine distance is 2 times of the Euclidean distance if you normalize the vectors in Euclidean distance situation, etc., etc. But now let's move on to the real data analysis. So we will illustrate the QC and all the metrics you can use. We can start by clean the O with the space. So make it clean. So later we will export our big X, the real matrix, to the space, rather than this small toy example. So my question for you is, have you found the accession number from this paper we can use? Normally, I just do a geo, they search this keyword, and then it will have several of this pop up. And I can see SGS, let's see SGSE. There are 11 of these. There are some of the reanalysis of publicly available. So this author of this original paper download a lot of other people's data to do the cross-validation or enhance their own study. But what I want to get is from, is that our paper? I want to get is the real GSE number from the author for this paper. So in the data availability section, you have this ID, which indicate this is the data, single-cell RNA-seq data generated by this study, has been deposited into the geo database under this. So this is the ID we need. However, from the interface of SC toolbox, we cannot use this directly because we are going to download each sample. So we need sample level accession number. So this number indicate the whole study. So this is study ID. And under each study, you may have 3 samples, you may have 4 samples, you may have 120 samples. So we have to click and visit this page. So this page has been made public in 2023, December. And then we can look at the summary and what study. This is the skin biopsy samples. And then from here, we can see, okay, there are 20 samples in this study. So this number, GSM, is what we need. So this is called sample level accession number. Okay, so we can select the ID and then know what sample we are going to download, put into MATLAB. In this study, they have different numbers of patients, 5 of them. So we have P5, 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 P5. For each sample, they have, for each patient, they have 4 different samples. So 1, 2, 3, from relational, this is a skin sample, and then also for the comparison, they have this non-relational samples. Okay, so you need to click in if you really want to download the data to make sure there are files under each sample. So when I look at this, I know, okay, authors have done a good job upload all the files that we need for the matrix, for the genes, for the barcode. And then I click the second, I check it also has files in there. If nothing here, we cannot use it. We have to find another way, maybe ask the author to email us a link to download from their hard drive somewhere. But once we see this, it's clear we can download them. So what I'm going to do in this small tutorial, we are going to download 2 samples together. So I will show you to download each one independently, then merge, because we are going to compare 2 samples. One from the same patient, one is diseased, and one is healthy skin. So we need this number first. Let's copy this number. I come back to the toolbox. Remember, we don't have SCE in this working space, so we don't use this. We just use this interface. Once the import data, let's select GEO database session number, okay? So this is my first. We can do this. We can put a 2 if you have 50. So you later I show you can do multiple accession number together. The program will automatically merge it for you. But I want to show you as independent. If you have 2 source of data, you prepare SCE for them, how you can merge them manually, okay? So once you have this sample, you can click and let the program to import the first. So once this is done, we are going to import the second. So the first ID and with a 449, we will then do the same thing for 450. You can repeat to get everything from this data set, 20 different samples all together put. It will be a huge data set. It requires large memory of the computer to hold that. So for the demonstration purpose, I only use first 2 samples to show you, but the rest of it will be the same. As long as you label the cell population, their source correctly, you should be able to identify what group of cells from which sample. Interface is okay, good. Yeah. shouldn't take too long, I hope. Oh, waiting, okay. That's a lot of cells. How many cells? I can't believe it. This is from this study. That's huge. Well, it's very well. That's a huge, yeah. That's the output, yes, from the download. That's a lot of cells. I can't believe it. Yeah. So you can go ahead and just do the filter because the filter will take some time. Yeah. Then the first one say okay. Then you just select. Yeah. Just do a filter to make it smaller. Yeah. Take some time.
So really, the input browser for loading the GEO numbers is just a command to get the software to go wget like those links, right?
Yeah.
Okay. That's
Yeah.
cool. It's a very good feature to have. Makes life easier.
That's right. So especially if you want to explore a lot of published data and very quickly, you don't do anything. You just use the interface to see, okay, there anything interesting by yourself.
So set it and forget it type of feature.
Yeah, yeah. This is a huge data set, so it's good examples to show the QC because we want to trim it by quality of cells, quality of genes to make it smaller. So today probably we cannot do the second sample because of the size of this. So we are going to focus on the first sample to trim it to do the QC correctly, okay? Well, normally we cannot get such huge number of cells. I don't know how they managed to do this. It's a weird situation anyway. So in this case, I want to check first. The data is correct. So I want to take a look of this first and see whether the genes are all with the name of correct human genes. Yeah, it looks like they are okay. They're all human genes. So I will first remove the empty genes. Empty genes means in so many cells, some rows, some of the genes, they all zero. So if you have a case like this, so this gene definitely should be removed first, right? We don't need them. There's no information unless you believe sample 2 have this highly expressed. But just for this purpose, we just remove empty genes. So this matrix contains a lot of empty genes. So after remove, we reduce the number of this to 22,000 genes rows. So there are quite a lot of 9,000 genes are empty in this huge data set. So we remove them. So this first step, just make this matrix smaller, okay? Oh, my computer crashed. So I'm going to use built-in small examples to show this anyway. Yeah, see you later. So now I'm going to use a smaller example data to illustrate the main steps you can use to trim off this to high quality set of data, rather than play with the raw data. If you have only one sample in such huge scale, then when you merge another one, it will definitely sooner or later blow up your PC. Unless you require the clusters. And by the way, the MATLAB should be able to run in clusters as well, but you may not have such nice interface. You may rely on the command lines to achieve some of the goals. So I'm going to use example data, it's much small, provided here to illustrate the major steps to do the QC. So I load this example data unprepared raw data. So this is much small. We can easily to handle. So as I said, you check whether the gene name makes sense. Okay. This gene names. and then remove empty genes first. Okay, there's no empty genes in my data, so nothing removed, so I keep everything intact. And then you can do, I will check, see here, I will check, select some genes expression. If you found the genes start with MT dash, MT dash. So those are the mitochondrial genes. So those genes are not encoded in nuclear genome, but they are in the organoids. This mitochondria has its own genome. There are 13 protein coding genes in there. So we can check the expression level of those genes. Say no, and then you can see the same plots. So I just quickly visualize to show you this my MT genes. They are highly expressed genes. For every single cell, there are a lot of mitochondria, if healthy cells. And also expression level for each of them are really high. You see for the ATP6, 100% cell has detected MT, ATP6 genes, so 100%. Then for individual genes expression, the highest can reach to 1,000 and some over 500 UMIs, and on average, still very high. They all abundant. So also, but you don't want to have significantly high proportion rates to map into MT genes, because they are, if you have excessively high level of MT genes expression, it indicates the cell has been lysis. The membrane is not, the mitochondrial gene membrane, mitochondrial membrane is not intact. That indicates the cell dies. So you want to have a QC which using the library size versus the MT gene rate ratio. So if you select this, it will generate a scatter plot for you to visualize these two statistics across all the cells. This will be a scatter plot. Yeah, so this is scatter plot. X-axis is the library size. As I said, library size indicates how many rates in total you found from a cell. So some of the cells you have many rates. So this could be two cells have been captured in one droplet. This may be a duplet, two cells in there, so it's almost doubled the regular library size you sequenced from that cell. So this may be not one cell, there are two cells. So somehow we need to remove this unusually high, highly sequenced droplet. So we set a cutoff. So you can select any cutoff. You don't want to throw too many cells, but you can see, okay, those ones are suspiciously duplets or triplets, maybe unusually high. You can set the cutoff here. If you're higher than this, I will remove. You keep all the regular cells in this range. You don't want it to be too small, so you have to have 500 or 1,000 the cutoff somewhere here. So there's no those cells has too small library size. If too small, indicate empty droplet. On your y-axis is how many mitochondrial rates have been mapped into how many rates have been mapped into the mitochondrial DNA genes. So this, you want to keep it small, okay, lower than 15%. So in this square, this window here, the cells we believe they're good enough. So if you want to set this cutoff lower, you can drag this bar down to, see, 11%, 10% if you're really stringent, if you want to get rid of the impact of this mitochondrial DNA genes impact in individual cells. So this data set has been already trimmed, so you see all the cells has lower than 15% of that. But your original, if you download directly this cutoff, there are some cells may have 80% or really high numbers, so you really want to set cutoff there. 15% is a reasonable number, but if you deal with, for example, the heart cell, and those cells, the muscle cells, those cells are notatiously known to be rich in mitochondrial DNAs. So you may want to relax this 15% maybe to the 20%, allow those cells to be well represented in your analysis downstream. Okay, we can see. Okay, we can learn how to drag this, reset. So somehow those values and the cells that will be removed will be highlighted in this small cross. And in this cutoff, I will exclude 68 cells. So only 3%, so not a big deal. So this set up this cutoff here, and then you're done. So you clean you remove 68 cells, but then nothing has been removed from the genes, so they only removed by the columns. Then you accept this, those cells will be removed. And you can come back and see, I want to look at the library size versus number of genes detected. So this is a scatter plot, same idea, but just change the y-axis to indicate the general patterns in terms of the distribution of cells in this space defined by library size and number of genes detected. So in general, if you have a cell, if you sequence it more, you should be able to detect more unique genes. That's why you see the clearly positive correlation between the library size and the number of genes detected. Okay, if you sequence extremely high, you should be able to detect for all the genes supposed to be expressed in that cell. Then at the end, you should see a plateau, and then the ceiling will be stopped somewhere, right? So this is almost reached that region. So we think that the library size is okay, and then this is relatively clean data. So if you have really messy data, you probably have some highly sequenced library here, but the number of genes dropped, and you really don't want those cells. You want to remove this too high, but keep the cell in the same region, range, okay? As I said, this is not a real good example because this data has been cleaned before. So, but I just illustrate the procedures. So if you use the cutoff here, you want to keep the detect gene number reasonably in this range, not higher, not too high, and library size is okay. So you get rid of 20 cells by this cutoff. You can remove them, then you remove another 20 cells. Okay? So I'm going to download using example data provided in the tool itself, rather than just found that huge data set from the publication. Just use this, but it's the same idea, okay? So I'm going to download this, the built-in pre-provided accession number for this sample, get some real data, and let's take a look the procedures of the QC. We will see the scatter plot and set up cutoff and trim the outliers. So this is the data we have. Let's do a filter first, remove empty genes. Okay, now the number of genes reduced from 300,000 to 250. This is the first step. Then we can start to look at the library size versus mitochondrial rates. That's a normal picture. You will see a lot of cells which have an extremely high mitochondrial rates because most of the rates have been mapped into mitochondria rather than more informative nuclear genes. Unless you are really studying the mitochondrial gene expression, you can keep that. But for now, we set a cutoff 15%. This will remove a lot of cells, and this library size, several cells have an extremely high level of this. So we can just click and get 15% here, 15%. Then we will remove 10% of cells, basically. Okay? Now we use this library size versus number of genes to look at this. It's a reasonable, good. We don't have to really do anything here. Okay, you expect this, and you reach to the 7,000, 8,000 genes uniquely detected in individual cells. This is really good numbers, and you sequence them deep enough. And the curves start to flat out already. So there's a limitation of how many individual genes you can detect from the single cell data, right? We really should consider the lower numbers here. So you may want to only keep 1,000 detected gene cells. So maybe rather than 0, we should set x-axis. Yeah, we can set the library size has to be 1,000, and then the y, the number of genes you detect has to be 1,000. So in this case, we will remove those cells which not very informative. The sequencing dips is low, number of genes only detect 900 genes, maybe not very useful. But you can set up this cutoff yourself as long as you believe it's reasonable and not impact your final results. You don't want to throw away a lot, but you don't want those empty droplets to compromise your results. So in this case, I remove 95 cells, accept this change. Okay. Another filter we can use is long non-coding RNAs versus gene numbers. This is not a very commonly used cutoff or QC step. But in reality, in practice, we realize that some of these long non-coding RNAs, although they are not supposed to be poly-A enriched, but they occupied a lot of the library rates. For some reason, for the combinations of those long non-coding RNAs, you have see these cells, for example, have quite a lot of those rates, which are not in the normal range. We don't know exactly what happened in there, but this is by empirical some evidence. If you remove those cells, will be good. So we keep this the same, but maybe we remove some of those long non-coding RNA genes, enriched cells. So I can move this up a little bit. I don't care the number, so I only cut through this. 19 cells will be removed. Accept change. Okay. There's a recent paper say if you don't have, so we remove MALA1 gene, that's in the category of long non-coding RNA. But in the opposite, if you have a cell which is supposed to have a lot of non-coding RNA, but you don't have this expression at all, there are also indication of this low quality. There's paper people found. Okay, if your cell in human mouse, if you don't have this MALA1 long non-coding RNA gene expression, it's also problematic. So I add this in as a knowledge-driven cutoff to remove those genes, have zero expression for MALA1 gene. Okay, because normally you don't expect to have that. So this will not define. Yeah, so so I may fix this problem later. Cannot do it for the sparse matrix. There are other options you can have if you want to make the data more informative. For example, if you do analysis, you don't want hemoglobin genes. You don't need ribosome protein coding genes because those genes are boring, basically. Okay, their function is clear, and you don't want to analyze them, mess up with other genes. There are options you can remove those. You can remove mitochondrial genes, although it's highly enriched in most of cells. But if you are not interested in this, you don't want to build a gene regulatory network, including mess up with mitochondrial genes, with the nuclear genes. So there are options, depends on your application downstream. You can remove, for example, you only want to use genes with official name. So when the gene has a special name, even you found it significant in some study, but you don't have interpretation because it's unknown genes. So in some case, you want to remove those genes to make your data is more focused. Let's see some examples of this without approved samples. So if you look at this gene list, so if you look at AS, the dash AS is antisense transcripts. So people know genes transcribed from one direction to another, like official transcription. People always found some long genes, you have a small fragment which go opposite direction, give you a transcription for some part of the genes. If you focus on those research, you may say, okay, this is important because it's create antisense transcripts maybe targeted to the parent genes and inhibit its expression somehow. But if you have no idea what they are, you can remove this from analysis. So this is one case, and there are quite a lot if you don't remove those antisense transcripts. So those genes, for example, FS, these RNA genes, you don't want to include them because they have a different transcription machinery. They are very different from protein coding genes, or you can remove them. At least you have a strong reason to keep them. So some genes, I don't know whether this officially named, but it's open reading frame genes. People just put annotation in there, but there's no function related. So a lot of this, okay. So you have option in this step to exclude them from analysis. So you see this, all this. So what are they? They're protein, ribosome protein genes, also different isoforms maybe. Yeah, so let's see if I remove those genes, remove this gene without approved symbols. This is, well, I don't know, this may be mouse, it's human data. Yeah, in this case, we will remove 8,000 genes. That's quite a lot. Now, if you look at your list of genes, it's better, right? Remove those stringent names of genes. So all these gene names are officially accepted. They have official name. You can also remove gene by name. So if you don't want to, gene looks like with AS, with linker genes, so those are long non-coding RNA genes, with any of the GM, RIK, so you can remove them. So if I do this filter, I remove another 1,000 genes. Now I have very, very clean genes set. Okay. Then you can use this basic QC basic function. In this case, we will clean cells and genes at the same time. So I have several cutoffs. You can customize the values. So I need library size at least 1,000. Okay, we already done this, so this will not have a huge impact. But because we changed the number of genes, so your whole matrix changes the row numbers. So this may be applicable because you remove a lot of junk genes with the special names. Now the library size may change. So we still want to keep some of this, and also the mitochondrial ratio. We want to have a cell have a minimum number of genes expressed 15, and minimum non-zero genes per cells 500. Then you can allow this program to do the iteration. It will first use the cutoff you set for the cells to remove cells, then remove genes based on your cutoff, and come back after you remove genes, your matrix becomes smaller, so you try remove cells again based on your cutoff until the whole procedure stopped. There's no cells or genes can be removed. In this case, this will be very clean data after you do this iteration. There are two options I gave. You can be more relaxed. Cutoff gave you more cells, more genes at the end. But then you can also select more stringent options to remove more cells and genes, give you cleaner process data at the end. So those are the options you can have. So I think we are done with the single data set QC and retrieval from a database. So we know how to deal with one. And next week, I think I will select a right size data set to show you how to combine if you have two data sets you want to compare at the end. But right now, we just stop here this week. Okay, so the whole procedure changed, and then I will remove 2069 genes and 4 cells. If I accept this, the change will be made, okay? And you have a really clean data. For this small example, you can view metadata. You will see, okay, this is from where you download. So because you have this accession number of the samples, you can download, you can revisit this page in the website to find the information about this sample. Okay, that's where the metadata is located. Okay, any questions before we finish this week? Okay. Yeah, question.
QC cutoff we choose is based on experience or something?
Yeah, it's based on experience, so you can change it if you like. Yeah.